Those of you who are just joining us, this is the International Eosinophil Society webinar. We're going to be starting in just another few seconds. I'm just waiting for everyone else to log in. So good morning, good afternoon, or good evening, depending on where in the world you're logging in from. So I want to welcome you to the International Eosinophil Society webinar. Today's webinar is going to be the 2025 Congress highlights. For those of you who were not lucky enough to join us in Montpellier last summer, we had an amazing conference spanning over 4 days at the south of France. There were some fabulous talks, and we thought we would share some of them with you today. So today is the webinar, the highlights. We have 3 wonderful speakers lined up for you this morning. First, a little bit about the International Eosinophil Society. We're an organization of scientists and clinicians who are interested in the eosinophil, which is an immune cell that is strongly associated with many diseases. The Society sponsors biennial meetings, monthly webinars, and other educational programming for its 300+ members to review new information and to share data about the eosinophil and its role in health and disease. And I should have introduced myself. I'm also Allison Fryer. I'm the president of the Eosinophil Society for the next 2 years. If you are not already a member of the Eosinophil Society, I urge you to join. We have put the QR code on the screen. And I want to tell all of the young scientists out there, if you're a pre-doctoral student, postdoctoral fellow, or resident, it is free for you to join the society. So please take advantage of this offer and become a member of us. If I can have the next slide. We are already beginning to plan for the next Congress, which will be in 2027, a year and a half away, and we're going to be holding this conference in the beautiful city of Vancouver in Canada. So please put this onto your calendars. It's the 12th to 16th of July, 2027. And the speakers of our next webinar, which is coming up in December, so this is December 10th, we have 3 wonderful speakers lined up to describe the hypereosinophilic syndrome overview and unmet needs. So again, please mark your calendars for December 10th. This is a Wednesday next month. And I would love to thank all of our corporate advisory councils. So please join me in thanking them: Platinum members AstraZeneca, GlaxoSmithKline; Gold members Amgen, Sanofi, Regeneron; and Bronze Celldex, Takeda, and Uniquity Bio. So thank you hugely to our corporate advisory council. We could not be putting these webinars on without their support. Next, I'd like to introduce my co-moderator this morning, Dr. Nicola Diny. Nicola is a junior group leader at the University of Bonn in Germany, and she studies eosinophil plasticity, tissue adaptation, and functions. Nicola.
Thank you, Allison. So just a few housekeeping rules. For any questions you have to the speakers about their talks, please use the question and answer button to ask. You can write your questions anytime, and we will read the questions to the speakers. Throughout the webinar, all attendees are muted and the chat feature is disabled. This webinar will be recorded and will be posted on the IES website. If you'd like to view closed captioning or live transcription during the webinar, please click the Show Captions button. Okay, and with that, it is my pleasure to introduce our speakers for today's webinar. We have our first speaker will be Christophe Desmet, who will speak on the regulation of the eosinophil lineage in eosinophil-associated diseases in the context of precision therapy. Our second speaker is Jennifer Felton, who will speak on the transcriptional and epigenetic regulation of eosinophil development, maturation, and tissue specificity, promoters, enhancers, and beyond. And our first and last speaker will be Leah Kottyan, who will speak on the relationship between genetic variants that regulate blood eosinophilia and allergic disease risk. So our first speaker for today's webinar, Christophe Desmet, is a molecular biologist at the Giga Institute of the University of Liège in Belgium. He specializes in the regulation of myeloid cell plasticity and function during hematopoiesis and within tissues, with a particular focus on eosinophils and their impact in the respiratory tract. And with that, please start your presentation on the regulation of the eosinophil lineage in eosinophil-associated diseases in the context of precision therapy.
Thank you very much for the kind introduction, Nicola. I'm trying to figure out where my presentation went now. Okay, here it is. Okay, you should see it now. All right, thank you very much for the kind invitation, for having me here, and hello to everyone. These are my disclosures. So most of us in this audience probably study eosinophils not because of beauty of these cells, but because they are associated with a lot of chronic diseases that we usually term now eosinophil-associated disorders, or EADs. And in these variety of diseases, eosinophils are either used as biomarkers, so they are present in the tissues in abnormal quantities associated with pathological features, but seem to be more like bystanders or not essential drivers, at least, of the disease. But in others, they accumulate in the tissue and they cause trouble in the tissue as well. And so they are causative agents in disease manifestations. And especially if you think of EADs that in which eosinophils are really bad, you think of eosinophilic asthma, or so-called type 2 asthma, and hypereosinophilic syndrome, or AGPA. So what happens in these disorders usually is that you have an underlying type 2 inflammation that becomes chronic. And the disease tissue will release alarming cytokines like interleukin-33 and TSLP that will activate type 2 lymphoid cells and make them spew out abnormal quantities of type 2 cytokines like interleukin-4/13 and interleukin-5. And most of you know that interleukin-5 is the favorite cytokine of eosinophils, right? And so what it will do, this IL-5, is that it will go to the bone marrow and it will instruct hematopoietic progenitors to produce more eosinophils. So you increase the output of eosinophils from the bone marrow, and this will be essential in sustaining blood eosinophilia. And then, of course, IL-5 also has other actions in eosinophils and in the tissue, notably it will increase their survival, contribute to their activation, and thereby contribute to the disorders. For interleukin-4 and interleukin-13, their role directly on eosinophils is less well understood, but what it seems to be really doing, these two cytokines, that they promote the recruitment of eosinophils inside the target tissues. Nowadays, we are lucky enough to have an increasing portfolio of monoclonal antibodies that help dampen specific arms of the type 2 inflammation. For instance, anti-TSLP antibodies, tezepelumab, that will block the alarmin signaling, or dupilumab, that will block interleukin-4/13 signaling. And we also have several molecules that target the addiction of eosinophils to interleukin-5. So one is benralizumab. It's an anti-interleukin-5 receptor alpha directed antibody that triggers antibody-dependent cell cytotoxicity, meaning this monoclonal antibody fully depletes the eosinophil lineage, so both the progenitors and the mature progeny. And then you also have antibodies that neutralize interleukin-5 itself, like mepolizumab and rituximab, that will decrease the output of the bone marrow. And today, our focus will be really on these two types of regions, so the anti-interleukin-5 regions that decrease the output of eosinophils. Because, well, it's very clear that they do that, right? So if you consider here this cohort of severe eosinophilic asthmatic patients that were eosinophilic in the beginning, when you give them anti-interleukin-5 treatment, they quickly become eosinopenic. So they still have residual eosinophils, but they are far below the median of the general population. So, but how does this decrease happen? What is happening to eosinophils? And historically, one of the first studies on mepolizumab in patients in vivo was performed by Judah Denberg's group, where they looked in the bone marrow of patients with asthma that were given anti-interleukin-5 antibodies, mepolizumab. And what they noticed is that the number of eosinophilic myelocytes and metamyelocytes would go down in the bone marrow of these patients. And what they concluded was that not having interleukin-5 would arrest the maturation of eosinophil progenitors. And this is why in all textbooks you will see that interleukin-5 promotes the maturation of eosinophils, right? So, but is that so? Because if you read different reviews, you'll find a flurry of potential activities of interleukin-5 on hematopoietic progenitors. Some say it will promote the eosinophilic fate from hematopoietic stem and progenitor cells. Others will say it just expands the lineage, and many will say it promotes the maturation of eosinophil progenitors. So together with two very talented PhD students, Joseph Jorsen and Glenn Valhils, we decided to really figure out what was really going on in vivo. And they didn't know at the start of the work, but this would lead them to get the Glyc Award of the IES. And I want to thank the IES again for this wonderful boost to their morale and career, and also for a wonderful congress where this prize was awarded. But let's get back to 22 something, where they started the work. They were not that happy, as you can see, because they ran into several issues. And they pertain to actually the ontogeny of eosinophils and the maturation pathway that was not very well resolved, in our opinion, and also lots of trouble with single-cell RNA sequencing, which most of you will know is very challenging with eosinophils. So they are challenging once they mature. But if you look just at the earliest stages of eosinophil development, single-cell RNA sequencing just works fine. Actually, here you have bone marrow progenitors from the mouse bone marrow. And you can see that you nicely can capture eosinophil progenitors using just regular 10x genomics. So they are labeled with PRG2, PRG3, and EPX. And in this picture, what is maybe striking you is that eosinophil progenitors are very close to basophil progenitors. That might be expected. But look how close they come to megakaryocytes and erythroid progenitors, whereas they're really far away from neutrophil progenitors. Actually, they gather with megakaryocyte and erythroid progenitors in this GATA1 arm here of the hematopoietic tree. And you will all know, and you will hear more of it in the second talk probably, that GATA1 is a transcription factor that is very important for the development of eosinophils. So they are here, remember that. Now, things get really bad when you want to move down the line. So for progenitors, single-cell RNA sequencing works fine. But here we try to figure out what we were losing, because we didn't get many mature eosinophils. We actually got very few using 10x genomics. So we thought use site-seq. So have the antibodies there in the mix as well that you can recover to maybe rescue cells that you would otherwise not see in the analysis. And by doing so, we can really capture a nice continuum of eosinophil development with progenitors here that express c-KIT, then downregulate it and start to upregulate CCR3 as expected. And you have here presumably mature eosinophils. So you have this continuum of maturation. But look at the RNA content. In the first early stages, you have a lot of RNA. But then as soon as you move down the maturation line, you lose most of your RNA and most of your cells become non-analyzable and RNA information is lost. So single-cell RNA sequencing to us was really the way to go to study eosinophil maturation. So we decided to go back to basics and go back to what most people do to study hematopoiesis. It's flow cytometry. And you're all familiar probably with Koichi Takashi's method for identifying eosinophil progenitors. In humans, eosinophil progenitors are found in the pool of common myeloid progenitors through their expression of interleukin-5 receptor alpha gene. So here are the EOPs. But then if you want to look at mature eosinophils, you will use a completely different gating strategy. And so you have a gating strategy for EOPs and another gating strategy for mature eosinophils. And so you miss a lot of what's happening in between. Okay, but that's a minor issue compared to what you will run into with mouse. So in mice, you're also familiar with this strategy where you will find EOPs as a subset of the granulocyte monocyte progenitors in mice. That is also labeled by interleukin-5 receptor alpha expression. But what you need to know is that in Akashi's original paper, they used a clone for CD125 for IL-5 array that's called H7. And that's not the clone that made it to the market. Actually, clone T21, another clone, was brought to the market. And this clone is really, really troublesome, to say the least. So here you have wild-type mice, and you have neutrophils and eosinophils labeled with 3 of the antibodies that are available to stain for IL-5 array. You will all know that T21 is a very good marker for neutrophils, right? It stains neutrophil far better than it stains eosinophils in the mouse. And you will also see that RIA343, that is sometimes taken as a control for T21, but it's actually the same antibody, just does the same. But look at this third clone, DIH37, stains eosinophil, but not neutrophils at all. So in the lab, we generated IL-5 array knockout mice by knocking in an EGFP reporter cassette inside the IL-5 array locus in frame with the coding sequence of IL-5 array. So in the homozygous state, these mice are fully deficient for IL-5 array. And look at what happens to T21 staining. It stays exactly the same, okay, in neutrophils, and the same for RIA343. So meaning these antibodies are specifically labeled neutrophil lineage cells. And if you use them to track EOPs or eosinophils, and especially for EOPs, you will have a lot of contamination by neutrophil lineage cells. So do not use these antibodies. You will do yourself a big service. So there was a problem of resolution and continuum for studying eosinophils using the classical strategies around inflow cytometry, and that was really annoying to us. But fortunately, around the time we ran into these issues, Etienne Becht described a really interesting technique that's called infinity flow. And that's a way of virtually staining single cells for hundreds of cell surface markers using just conventional flow cytometry. So it's really a great technique. So how do you implement it? So in our case, it was to study eosinophil development, right? So what we would do is we would take bone marrow from human or mice, get rid of some lineages we were not interested in using dump staining, and we would label all these cells with a backbone panel of antibodies that we really carefully design. So if you use infinity flow, the backbone is the most important part. So you need to take time to really optimize it. So what you need to have in this panel is markers for lineages of interest, but also markers that you expect to be dynamically regulated in your process. So in our case, all the markers in green here were known to be expressed at some point in the eosinophil lineage, and some of them were dynamically regulated, and it was already known, like KIT is in the progenitors, and CD11B is higher in the mature eosinophils. So we included them, obviously, in the backbone. And in mice, we added another marker that is called CD55, and that's a very nice marker for GATA1 lineages. So GATA1 again. So now that you have labeled all your cells with this backbone panel, you ventilate them into 96 well plates, in which in each well you have a separate antibody to a specific cell surface marker that is coupled to just one fluorochrome, a strong fluorochrome, usually PE. And you run all these wells through classical flow cytometry. And what you get out of this is a huge amount of data that you can arrange in a porous matrix where you have all the cells labeled with the backbone antibody, but then batches of cells that correspond to a well in which you only have one antibody and one signal. So, oops, here, like this one, for instance. But because the backbone is present in all the wells and the same populations of cells are present in all the wells too, you can infer what the staining would be in all the other wells. And doing so, you can complete the matrix and get really something super resolved, let's say, because you get hundreds of cell surface markers per cell. And if you do that and look at a picture of hematopoiesis in human or murine bone marrow, you see that you get a really nice picture. So hematopoietic stem and progenitor cells in human are here, and here you have eosinophils coming out of the HSPC pool, and you can follow fully the maturation towards the mature eosinophil stage here. And in the mouse, the conformation of this UMAP is even easier to follow. You have the pool of hematopoietic stem and progenitor cells here, and then you have this long arm of myelopoiesis, along which you will find erythroid progenitors, megakaryocyte progenitors, basophil mast cell progenitors, and then in the end, eosinophil progenitors. So actually, this is already a bit the picture I showed you in single-cell RNA sequencing. And if you look in the literature, the recent literature on how hematopoiesis is organized in both mice and human, you will see that it's organized into two major developmental pathways. One is marked by the expression of GATA1 in the progenitors, so hence my reference to GATA1 lineages, and it's marked by CD55 expression at the surface level in mice. And in these lineages, we'll have eosinophils, basophils, platelets, and erythrocytes. And then you have a completely separate arm, which will give rise to macrophages and neutrophils. So actually, eosinophils and neutrophils are much further away, developmentally speaking, than is usually assumed. So, of course, you cannot run infinity flow on a daily basis. So you need to find a way to make things simpler. And Joseph and Glenn spent quite some time optimizing a reduced flow cytometric panel with select markers that would allow to really capture eosinophil development from its inception to the fully mature eosinophil stage using just 8-9 colors. So really doable in classical flow cytometry. And we divided the developmental pathway into 4 stages based on density nodes that we saw were quite well conserved across individuals and conditions in both mice and human. But of course, it's a continuum, right? But it was handy to cut it into stages like this. And oops, you can see that you have a nice progression from myelocytes in the beginning towards mature eosinophils in the end. And this is the general strategy. If you want to follow it, I invite you to read the paper. It's very easy to translate to your own laboratory. So finally, Joseph and Glenn, now with these tools in hand, were able to follow eosinophil development and answer the question, what does interleukin-5 really do to eosinophil development? So first, they looked at a model of strong eosinophilia. So they induced eosinophilia in mice by injecting every day recombinant interleukin-33. And within 7 days, the mice are packed with eosinophils. And if you look in their bone marrow, you will see that in the first days of stimulation, the mature eosinophils will go out of the bone marrow. But then, starting on day 3 to 4, you will start to have a massive expansion of the early stages of myelopoiesis. And then mature eosinophils will start to accumulate. And then as soon as you stop the stimulation, things will go back to normal. So you see how dynamic the lineage can be. Now, what happens if there's no interleukin-5 around? So if you inject interleukin-33, you get massive amounts of interleukin-5. So you expand the lineage. But what happens if you don't have interleukin-5? Does the lineage arrest? The answer is no. Actually, all the stages of eosinophil development are still present in IL-5 deficient mice. But what happens is that the lineage flatlines. So you can see the blue dots here, sorry. In the steady state, the gray squares, you have a progressive expansion of the eosinophil lineage along the maturation pathway. But in IL-5 deficient mice, it completely flatlines. And even if you boost the mice with interleukin-33 and try to get things moving, let's say, there's still no interleukin-5 to sustain eosinophilopiesis, and the lineage still flatlines. So there's no maturational arrest and no decrease of eosinophil progenitors. So this is quite normal. Next, Joseph and Glenn looked at cell cycling activity in the eosinophil progenitors in IL-5 competent and IL-5 deficient mice. They used incorporation of base analog of DNA. So actually what you monitor here is DNA synthesis. And it's just a 1-hour pulse in vivo. So that's very short time window. But within this very short time window, 50% of myelocytes in wild-type mice would capture EDU incorporated in their DNA. So you see how fast the cycle goes. But in IL-5 deficient mice, the picture remains exactly the same, a little bit less maybe, but still cells are pretty much able to cycle, even though a bit less in the stage 2. And you can see that in stage 3 and 4 cells, which look like metamyelocytes and mature eosinophils, you don't have any incorporation anymore. What's interesting is to look into the IL-33 model of eosinophilia. In that case, the picture becomes quite different. You see first that the percentage of EDU-positive cells within an hour in the myelocyte gates becomes really high. So it's much higher even than in the steady state, around 3/4 of the cells have incorporated EDU within an hour. And interestingly, even in stage 3, that normally has stopped proliferating altogether in steady state mice, you still have cells that incorporate EDU. And so meaning interleukin-5 looks like it's promoting something that's called transit amplification. Oh yeah, by the way, in interleukin-5 deficient mice, you don't see that. So showing really interleukin-5 is responsible for this. So what is transit amplification? It's a rather common process in developmental biology, actually, and hematopoiesis. It's very well studied. So I cite this paper very often because it nicely shows how the process goes. So hematopoiesis can be seen as a balancing act of progenitors between cycling, so dividing themselves, and maturing to give rise to the mature progeny. And if you can play with this balance and slow down the maturation and give more room for cycling, you have more time for the cells to divide, and so you will increase the output of cells of this lineage. And this is what happens with glucocorticoids in hematopoiesis, for instance. And we have exactly the same happening in eosinophils, actually. So using transcriptomics here, so we analyzed eosinophil progenitors in the bone marrow of mice treated for 7 days with interleukin-33. Here you have a steady state, and here, so this strong model of eosinophilia. That looks first at the transcriptome of stage 1, 2, 3, and 4 eosinophils in the control condition. You see that genes associated with maturation, effector functions of eosinophils, go up mostly at stage 3, whereas the genes associated with cell cycling go down at the same time. Now look at what happens in IL-33 treated conditions. In stage 3, the maturation genes are just slightly upregulated. They get upregulated later. And the opposite is true for the cell cycle associated genes. They stay active, upregulated longer, actually. So really mimicking what I just showed you. You have less maturation and more cycling. And you can even make the point that this is interleukin-5 dependent. So after, in this case, we treated the mice that received interleukin-33 with anti-interleukin-5 antibodies at the peak of eosinophilia. And we analyzed these mice 3 days later. And you can see that you get the opposite picture. So with anti-interleukin-5 antibodies, you downregulate the expression of cell cycle associated genes quicker, and you upregulate genes associated with maturation faster. So one of the take-home messages of the work of Glenn and Joseph is that interleukin-5 is actually a rheostat for the transit amplification of eosinophil progenitors. When you have a lot of interleukin-5 around, you will boost the cycling activity of eosinophil progenitors and delay their maturation. So remember this. So interleukin-5 is not a promoter of eosinophil maturation. It's a brake to eosinophil maturation. And this is why ex vivo, when you make eosinophils with recombinant interleukin-5 on bone marrow cells from mice, it takes almost 2 weeks. It's not a fast process. It's very slow. It's because it's just the expansion you're looking for. And by blocking interleukin-5, you actually really downregulate this process. You downregulate cycling, you accelerate maturation, and in this way, your patient becomes eosinopenic. So now I hope I could convince you that interleukin-5 centrally is actually a regulator of the transit amplification of eosinophils mostly, and this is how it regulates the number of eosinophils systemically. But it might also have other effects, and we need to look into what happens to eosinophils that are exposed to high levels of interleukin-5 or are completely depleted of interleukin-5 at the functional levels, notably in tissues. And with that, I would like to thank all the people involved in the work and you for your attention. And I encourage you to have a look at the full publication of Glenn and Joseph, because it contains much more information than I had time to share with you today. And with that, I will be happy to take questions.
Thank you very much for this great talk, Christophe. I don't see any questions yet in the Q&A box. So to all our attendees, please type your questions in. And in the meantime, maybe I could start. I was wondering, so in your IL-33 treated mice, you talk about how the increased cycling is still dependent on IL-5. So how do you think, is there a crosstalk between these cytokine signals in the bone marrow itself?
What do you mean, between interleukin-33 and interleukin-5? Is that what you mean?
Yes, presumably IL-33 treatment would not increase IL-5 levels in the mouse, right?
Oh, yes, it does. It does. So actually, the main effect of interleukin-33 is that it's a direct activator of ILC2s and Th2 cells. So it really promotes their production of interleukin-5. And so this is why it's much more efficient to give IL-33 to a mouse than recombinant interleukin-5, because you turn the mouse into a factory for interleukin-5, which is much more efficient than giving the recombinant cytokine, actually. Yeah, but it's indirect. In our hands, it's mostly indirect. So you can give IL-33 in the nose, intraperitoneally, you get the same effect on the central eosinophilia. The expansion is massive. Yeah.
So you don't think there's any direct effect of IL-33 on eosinophil progenitors?
It might be so, but we haven't seen anything conclusive, I would say. You have to know that interleukin-33 is quite toxic to the bone marrow, and so it gets neutralized quite efficiently through soluble receptors in the serum, and so it's hard to get a lot of IL-33 to talk to directly to bone marrow progenitors. But I don't exclude it might happen. But at least when we remove interleukin-5, it's like we have a wild-type mouse, so or IL-5 deficient mouse, let's say. So it doesn't look like we got a big effect of IL-33 itself. Yeah.
Thank you. We have a question from Stephen Ackerman asking whether you've seen any differences in the transient amplification of eosinophil progenitors, either in mouse versus human.
So that's a very, very interesting question, and that's one we're trying to address with Florence Rufus at the moment, looking at hypereosinophilic syndrome patients. Because in the mouse, we have a very dynamic system where we can control when we give the interleukin-5 boost, let's say, but in humans, we don't have that. But it's interesting to look at the bone marrow of hypereosinophilic syndrome patients, because you would expect all of them have a massive expansion of the lineage and progenitors, but it's not the case. So in some patients, you have expansion of progenitors. In others, you only seem to accumulate mature eosinophils. And we don't know if it's just a matter of looking at the right time. So maybe in a patient, we're just looking at the conclusion of boost of interleukin-5, or if it's really a difference in the way the lineage is regulated across patients. We don't know. We don't know that for now. That's a very, very interesting question indeed.
Okay, thanks. And I think we have another question from Judah Denberg, who thanks you for the excellent distinction between transient amplification and maturation, and whether you think that there are other cell cycle specific processes that IL-5 can affect.
What does he mean by, I don't know what he means exactly by cell cycle processes, but it's interesting to look at the transcriptomic signatures, actually, because it's just not cell cycling that interleukin-5 seems to be boosting. So that's why we say there might be other effects as well that we need to address, not through transcriptomics, but maybe through metabolomics. Yes, so for sure, cell cycling is so intertwined with metabolics and the general metabolism in the cells. So there's really a lot to be done there. Yes, indeed. Yeah.
Okay. And I think this is maybe a follow-up or a similar question on the concurrent role of IL-3 on this IL-5 induced process. So maybe the distinction of the two cytokines, or what IL-3 plays in this process.
Yes, so that's very interesting. So for the expansion, it looks really like interleukin-5 doing all the job, because when you don't have interleukin-5, you cannot expand the lineage, that's for sure. So eosinophils really rely on this one to expand. But about commitment, for instance, and baseline proliferation, let's say, there is room for other regulators, and maybe interleukin-3 and GM-CSF and maybe other ones as well are important, because you've seen that with the EDU and cell cycling, right? So even if you don't have interleukin-5, your early stages keep proliferating. So there is more to the commitment, the expansion than interleukin-5, indeed.
Thank you. We have another question from Mate Goya. Whether these eosinophils that with delayed maturation, whether they end up functionally distinct.
That's really one of the questions we wanted to address. So yeah, we're looking into this at the moment. Yes. We haven't seen anything yet, but we're looking at just really raw things usually, right? Just recruitment into the tissues, and we need more granularity, I would say, there, more details. So we're gonna keep digging. But the cells undergo really dramatic changes. Even if you look at them morphologically speaking, you see that they become bigger in the mouse. It's very striking, and so you would expect that the progeny will maybe be a bit different, but we don't have evidence for that yet.
Okay, thank you. I think we have time for one more question. We have a question here from Timothy Hart, who's asking about the effect of dosing recombinant human IL-2. Sorry, the effect of dosing recombinant human IL-2 in monkeys leads to a more rapid eosinophilia than it appears to have occurred in the mice with the alpha-D3 treatment. Could there be differences in your interpretations depending on the stimulating cytokine, or if it's a natural stimulation like a parasitic infection?
Yes, so it's obviously a very artificial model. We're using a recombinant cytokine, interleukin-33. That's an alarmin. So we still think it models quite well what happens, for instance, in asthma, where interleukin-33 is one of the drivers of eosinophilia. But it's true, there's not just one way to roam. Anything that will boost interleukin-5 production will boost eosinophil output from the bone marrow. And I would expect that depending on the species, you could have some stimuli that are even more potent than interleukin-33 at stimulating type 2 cells or ILC2s into producing interleukin-5. Yes, yeah.
Okay.
But I don't have a definitive answer for the question, for the specific question.
Thank you very much. There's another question, but I think we'll have to move on. Maybe you could answer it in the Q&A box.
Yes, I will have a look. All right.
Thank you
Thank you very much.
Okay, thank you all for this lively discussion. I would now like to introduce our second speaker for today. Jennifer Felton is a senior research associate in the Rothenberg Cured Lab at Cincinnati Children's Hospital. Her work focuses on investigating the transcriptional and epigenetic factors that influence the processes of eosinophil development and tissue specificity, primarily in the esophagus, in eosinophilic esophagitis, and in the gastrointestinal tract. And today, Jennifer Felton will be speaking about transcriptional and epigenetic regulation of eosinophil development, maturation, and tissue specificity, promoters, enhancers, and beyond. Thank you, Jenny.
Thank you very much for the kind introduction. Let me just share my slides. I would like to thank the organisers for inviting me back to give a more detailed presentation that I gave in France earlier in the summer. As I mentioned in Montpellier, this work is a culmination of many collaborative endeavours between members of the society, which has been initiated through the conference itself. So I'm really glad to be here again. Most of what we know about regulation of eosinophil lineage commitment and development is primarily understood in the context of the bone marrow. And we know this process is orchestrated by the dynamic regulation of transcription factors in the process of cell fate commitment. Eosinophil lineage commitment is primarily dictated by a number of transcription factors, including members of the CBP isoforms GATA1 and PU1. And the expression of these transcription factors is fine-tuned by their interactions with other transcription factors, for example, FOG1, friend of GATA, which regulates GATA1 levels. We see the importance of GATA1 in expression of many of the important eosinophil granule proteins, as well as CCR3 and IL-5 receptor alpha. As the eosinophil progenitor, the EOP, or the eosinophil committed cell, becomes a mature eosinophil, we see increase of transcription factors such as XBP1 and ID2, which promote survival during the demanding maturation and granulopoietic process. Whether there's dysregulation in the order or the expression level of one or more of these transcription factors has dramatic impacts on eosinophil production and lineage commitment. For example, if we force express GATA1 prior to CBP alpha expression in a progenitor cell, this actually drives basophil lineage commitment and completely prevents generation of an eosinophil committed progenitor or a mature eosinophil. In contrast, loss of CBP epsilon or XBP1 during this terminal differentiation process really impacts mature eosinophil development, but has no impact on eosinophil lineage commitment. So we can see the importance and the importance that the order of these transcription factors has in this elegant paper from 2021 from Steve Ackerman's lab, where they used CHIP qPCR to identify transcription factors that were bound within the promoter region of the IL-5 receptor alpha. And they did this using a 21-day eosinophil differentiation model from CD34 positive progenitor cells. And what you can clearly see from these graphs is that the IL-5 receptor alpha promoter was subject to distinct temporal regulation by discrete transcription factor families. First, at day 7, we see an increase in binding of PU1. Following this, at day 14, the CBP isoforms alpha and beta start to become upregulated, as well as PU1. And then finally, by day 21, when the final maturation occurs, CBP epsilon is very highly upregulated. This shows that there is dynamic changes in the transcription factor landscape in a developing eosinophil, and the sequence and the order of this transcription factor expression is essential. So how can we interrogate the transcriptional and epigenetic regulation of eosinophils? Christophe spoke a lot about the development of single-cell RNA sequencing in eosinophil populations, and we know that this can be commonly done with either gravity-based technologies such as the BD Rhapsody or other fluidic-based systems like the 10X. And there's a number of publications that have come out in recent years that are really focusing on utilizing single-cell RNA sequencing in peripheral blood eosinophils, as well as those from patients with nasal polyps or from tissue eosinophil subsets in mice, such as the small intestine and the esophagus. But DNA sequencing technologies are broadly split into categories related to the accessibility and the modifications associated with DNA. The first is ATAC sequencing, which relies on the openness or the availability of chromatin. Our lab has successfully managed to develop techniques to perform ATAC sequencing on bone marrow eosinophils, as well as FACS-sorted tissue eosinophils from the bone marrow, lung, and esophagus, as well as isolated human peripheral blood granulocytes. Typically, this technique requires around 50 to 100,000 eosinophils per replicate, but due to the epigenetic regulation of eosinophils and their transcriptional modifications, we often need much larger numbers, so closer to the 75 to 100,000 level. There are a number of platforms which have recently been developed that can offer single-cell ATAC sequencing, and this can often be paired or performed at the same time as single-cell RNA sequencing, so you get both the epigenetic and the transcriptional data from the same samples. In contrast, chip sequencing is really concerned with the chromatin structure, whether that's post-translational modifications to the histone tails, DNA-associated proteins, or transcription factors. Chip sequencing requires a much larger number of cells, typically 500,000 to 1 million, and we've successfully been able to perform this on FACS-sorted bone marrow eosinophils, both mature eosinophils and progenitors, as well as BMEOs and isolated peripheral blood granulocytes. Unfortunately, we've not been able to perform successful chip sequencing on FACS-sorted tissue eosinophils, but this is more due to a cell yield, like a cell number issue, rather than a biological reason, mainly because it's very difficult to get the required number of cells, 500,000, from FACS-sorted tissue digests from mice, even if you're pooling multiple samples. And this is even more difficult in human biopsies. Whilst we've been developing these technologies in our lab, we've developed a number of tips which have really allowed for successful DNA sequencing on eosinophils. Firstly, as I mentioned previously, scaling up the cell numbers, especially in ATAC sequencing, instead of using 50,000, ideally you should be using closer to the 75, 100,000. And this really helps with getting good quality cells that you can visualize using sequencing. In addition, FACS sorting cells into small quantities and pooling replicates helps with maintaining the DNA structure, especially if you're having to batch multiple experiments to get to that higher cell number. Additionally, increasing the centrifuge times helps a lot, especially if we're pelleting a small number of cells, say 30 to 50,000 eosinophils for ATAC sequencing. Sometimes increasing the centrifugation time up to 10 or 15 minutes really helps to get a visible pellet that you're able to accurately pipette during the methodology. And then something that we're now using routinely in all of our DNA and RNA sequencing techniques, especially when we're doing enzymatic digest of a tissue, is adding a number of goodies. And these include DNases, protease inhibitors, as well as caspase inhibitors such as QVD-OPH. So how can we utilize these techniques to identify the epigenetic landscape? Studies have shown that specific histone modifications, whether they be methylation, as shown here in red, or acetylation in purple, are associated with different functional states. So methylation of DNA and histones causes the nucleosomes to pack really tightly together, which means the transcriptional machinery and transcription factors are not able to access the DNA and regulate gene expression. However, in contrast, acetylation opens up the chromatin landscape, facilitating transcription factors to be able to access the DNA and regulate gene expression. So here I'm showing you a representation of the epigenetics landscape of a promoter in an eosinophil-associated gene. I've in the red box, which is highlighted, you can see the schema on the right, which just amplifies what we're visualizing in that box. But the typical profile of a promoter is an open region of chromatin, as identified by ATAC sequencing, shown in blue, and very high levels of histone 3 lysine 4 monomethylation in green, surrounded by an area of histone 3 lysine 4 monomethylation, sorry. And it's this inverse relationship between the tri and monomethylation that really allows us to identify that this is a promoter. In contrast, active enhancers, which are often associated great distances from the transcriptional start site of genes, have these open regions of chromatin as identified by ATAC sequencing, but have high levels of monomethylation versus trimethylation. And it's this inverted relationship between promoters and enhancers, which allows their differential identification. We know that active enhancers are small regions of DNA that can actively regulate transcription independent of the promoter. And there have been a number of GWAS and EQTL studies that have shown the presence of disease-associated SNPs within cell type-specific active enhancers. Specifically, in the case of the eosinophil, there's been a number of AP1-associated SNPs linked to eosinophil-specific enhancers that could impact allergic disease. Furthermore, when we look at the epigenetic landscape of an eosinophil, whether it's following an allergy induction, for example, on the left here, we show bone marrow eosinophils, which have been ATAC sequencing has been performed on either in the control condition PBS or following house dust mite challenge, versus tissue-specific populations under the same inflammatory treatment, such as bone marrow eosinophils and esophageal eosinophils following HGM treatment. We can start to see that there are a number of shared and unique epigenetically regulated regions which differentiate the inflammatory stimuli and the tissue-specific phenotype. So this shows that there is discrete epigenetic regulation of eosinophils dependent not only on their inflammatory stimuli, but also their tissue location. So what are the distinct processes and what are the transcription factors that are potentially regulating these processes, and how are they different to those that are involved in eosinophil poiesis? So as they mentioned in our introduction, our lab is specifically interested in looking at eosinophils in the esophagus, especially in the context of EOE. So we generated the hypothesis that esophageal eosinophil phenotype is driven by their epigenetic landscape, which regulates their transcriptional expression by select transcription factor families. So to investigate this, we did a combination of single-cell sequencing techniques, including RNA sequencing, ATAC sequencing, and flow cytometry, to visualize the distinct esophageal phenotype. First off, we FACS sorted bone marrow eosinophils and esophageal eosinophils from the esophagus of IL-13 transgenic EOE mice. We then performed ATAC sequencing and identified over 30,000 genomic regions that were epigenetically regulated in esophageal eosinophils compared to the bone marrow eosinophils. An example of some of the key genes are shown at the bottom of the slide. PGR3, which is the precursor to the MBP gene, is preferentially enriched in bone marrow-derived eosinophils. You can see this by the increase in the openness of the chromatin around the transcriptional start site in the bone marrow eosinophils, which is shown in blue. In contrast, alleles, IKZF3 and retinol A, both of which our lab has shown to be involved in tissue-specific eosinophil functions, were more open and more enriched in the transcriptional start site around esophageal eosinophils compared to bone marrow eosinophils. So what genes and gene pathways do this massive number of epigenetically regulated regions relate to? We took the 30,000 regions and annotated them based on the closest gene and found there was a massive enrichment in esophageal eosinophils for gene pathways that were associated with inflammation, response to the external environment, regulation of biosynthesis, and intracellular signaling pathways. In contrast, the bone marrow-enriched epigenetically regulated regions were more concerned with eosinophil cellular structure and development. So this suggests that eosinophils residing in the esophagus are readily distinguishable from those residing in the bone marrow. So how is this epigenetic modification affecting the transcriptional profile of esophageal eosinophils? And to answer this question, we performed single-cell RNA sequencing on bulk esophageal tissue from both the IL-5 and IL-13 transgenic EoE mice. You can clearly see in the UMAP that we're able to visualize eosinophils using this gravity-based single-cell sequencing technology. In addition, we see many structural and immune cells that are also present in the esophagus in an inflammatory model. When we looked at the genes that were enriched in the esophageal eosinophils, we found nearly 1,700 genes that were enriched for pathways, again, related to immunoregulation, biosynthesis, and response to the extracellular environment, suggesting that the epigenetic regulation we're seeing from the ATAC sequencing is having a direct effect on the transcriptional landscape of these esophageal eosinophils. So next, we wanted to look at the immunophenotype. To do this, we developed a 17-color spectral flow cytometry panel where we took eosinophils from different tissue locations in the same mouse, testing both hemopoietic compartments, including the blood and the bone marrow, as well as tissue-specific populations, including the esophagus, the lung, and 2 portions of the small intestine, the duodenum and the ileum. And what you can clearly see from this heat map is that each tissue population of eosinophils has a unique immunophenotype. Specifically, in the case of the esophagus, we see enrichment of the complement receptor C3AR1, which we found to be unique to esophageal eosinophils, at least in our mouse models. This suggests that the esophageal eosinophil phenotype is distinct from the lung and the small intestine, and that each tissue population of eosinophils has a unique or combination of surface markers that are distinct based on their tissue environment. So we're seeing epigenetic regulation having an effect on the transcriptional landscape of the cells, which is directly impacting their surface immunophenotype. So what's regulating this? To get to the crux of the issue, we compared our single-cell RNA sequencing data set with our ATAC-seq data set to find the genes which we defined as the esophageal eosinophil epitranscriptome. And these were 761 genes that were both epigenetically and transcriptionally enriched in esophageal eosinophils. We then used HOMA, which is a putative transcription factor discovery program, which looks for the presence of known transcription factor binding site motifs within the epigenetically enriched peaks. And what we found was that there was a massive enrichment of AP1-associated transcription factor family members. You can clearly see in this bar chart that the red bars being the esophageal eosinophils, a massive enrichment of many of the AP1 family transcription factors. And when we used a string interaction database to look at the interplay between these transcription factors, we found that there was a very strong core complex of AP1 family members that were interacting and potentially driving this esophageal phenotype. In contrast, when we looked at the bone marrow eosinophils, we could see many transcription factor families shown in blue that are preferentially enriched during eosinophil poiesis. We see the CBP isoforms as well as the IRFs. So this is really demonstrating to us that the tissue-specific transcription factors are distinct from those involved in eosinophil poiesis. And when we looked at the presence of this core complex of AP1-associated transcription factors within either the esophageal or the bone marrow enriched regions, we could see that over 35% of the esophageal enriched regions had all members of this core AP1 complex. In contrast, the bone marrow, less than 5% of the regions had this core complex, suggesting that there really is direct interactions between multiple AP1 members regulating this process. However, we were primarily interested in one member of this family, ATF3. And this is primarily because we see a massive increase in the transcriptional expression of the ATF3 transcription factor in esophageal eosinophils compared to bone marrow eosinophils. So to investigate the role of ATF3 in esophageal eosinophil phenotype, we collaborated with Dr. Quinlan and Dr. Pasala from Australia, and this is a direct collaboration that was a consequence of the 2023 IES meeting in Canada, and they were able to transfect and generate for us ATF3 knockout EL1 cells. We have previously developed a method in our lab where you can either co-culture eosinophils or eosinophil-like cells such as EOL1s with esophageal epithelial cells, EPC2s, to drive the esophageal eosinophil phenotype. And when we repeated this co-culture experiment using the ATF3 deficient EOL1 cells, we found that over 45% of the genes that were associated with the esophageal epitranscriptome were differentially regulated in the ATF3 deficient mice, suggesting that ATF3 is having a profound impact on the transcriptional landscape of eosinophils. So we took this in vivo and we generated an ATF3 deficient eosinophil mouse model using the EOCRE system, and then induced esophageal eosinophilia following 4 weeks of allergen challenge with Alternaria. And the most striking thing we saw was that there was an increase in the number of eosinophils in the ATF3 deficient mice. And when we performed single-cell RNA sequencing on the same animals, we could clearly identify a population of eosinophils that were present within the esophageal tissue. Again, we're seeing the structural cells and other immune cell populations, which are having an impact on the inflammatory environment in the tissue. So we subset the esophageal eosinophils from the wild type and ATF3 deficient allergen-treated mice, and we found that and then we paired the differentially regulated genes with the 761 members of the esophageal eosinophil epitranscriptome, and we found that about 10% of them were upregulated in the ATF3 EOCRE allergen-treated mice. Most notably, one that was very significantly upregulated at a transcriptional level was the complement receptor C3AR1, which we'd previously identified to be esophageal eosinophil-specific immunomarker. And when we looked at both a proteomic level and the transcriptional level, we could see an increase in the expression of the complement receptor on the esophageal eosinophils from the ATF3 deficient mice, highlighting that ATF3 is able to operate as a regulator of eosinophil gene expression and esophageal eosinophil phenotype. So I hope I've been able to show you that we can now accurately describe the esophageal landscape at an epigenetic level, and that eosinophils residing in the esophagus are readily distinguishable from those in the bone marrow. We know that the epigenetic landscape informs the transcriptional phenotype of esophageal eosinophils, and esophageal eosinophils are enriched for pathways associated with extracellular sensing, biosynthesis, and the response to the environment. We also showed that every tissue population of eosinophils has a unique phenotype, which is driven by the epigenetic regulation of their transcriptional phenotype, leading to proteomic cell surface changes. We also show that there is a member of the AP1 family which are involved in directing this process, suggesting that the transcription factors involved in tissue phenotype and function are distinct from those involved in eosinophil poiesis. And we show, at least in part, that ATF3 may be responsible for regulating this process of gene expression and cellular phenotype in the esophagus. So this brings us back to the big picture. We're now able to combine our data set with those generated by others, such as Claire Mesnel and Nicola Diny's group, showing lung-specific eosinophils, and Elizabeth Arnold looking at the small intestine, showing that there are discrete proteomic, transcriptional, and epigenetic landscapes for different tissue-specific populations of eosinophils. And we're currently working on pairing analysis between lung and esophageal or bone marrow eosinophils from the same inflammatory model to show how an inflammatory stimuli may drive bipolar or differential transcriptional phenotypes under the same conditions. I would like to thank the organizers again for inviting me to speak, my PI, Mark Rothenberg, and everybody that's been involved in this project. Thank you very much, and I'm happy to take any questions.
Thank you very much, Jenny, for this great talk. We don't have any questions in the Q&A yet, so please don't hesitate to type your questions in. In the meantime, maybe I can start. So you showed how the combination of chromatin landscape with gene expression data can allow you to identify transcription factors, and then you followed up on ATF3. Were you able to, with this, basically predict other sets of transcription factors that regulate maybe distinct subset of genes?
Yes, so we did look at the presence of transcription factor binding motifs within the core 761 genes. There was massive enrichment for other members, so like KLF7 and other AP1 specific family members. We've also previously shown that PU1 is very highly enriched in eosinophil-specific superenhancers, and PU1 definitely came up out of our analysis. But there does seem to be a tissue-specific transcription factor repertoire. So we need more data to really answer this for certain genes. So we've not looked in the small intestine, for example, to see what might be driving like the CLEX or other small intestine specific genes.
I think we have time for one more quick question from Stephen Ackerman. He asks, I recall that there are AP1 binding sites in the IL-5 receptor alpha promoter. Any data on how this transcription factor family affects IL-5 receptor expression differentially in different tissues?
I've not looked at this specifically, but I know that we do see some changes in the expression of the IL-5 receptor alpha in our ATF3 deficient mice. It wasn't quite as significant as we saw in some of the esophageal specific markers, for example, the complement receptor. But I think this is primarily because we see low levels of the IL-5 receptor alpha in the esophageal tissue itself. It seems to be downregulated in that environment. But that's definitely something that would be interesting to look at, especially with the different isoforms of the IL-5 receptor.
Thank you very much, Jenny.
Thank you very much. It's my pleasure to introduce our third speaker today, Dr. Leah Kottyan. Dr. Kottyan is a professor at Cincinnati Children's Hospital Medical Center and the University of Cincinnati. Her research is focused on the genetic etiology of immune diseases such as eosinophilic esophagitis and systemic lupus erythematosus. Dr. Kottyan is also the leader of the Cincinnati Children's Basic Science Research Committee, which represents hundreds of faculty members conducting foundational research into new biological insights. Her research includes the discovery of genotype-dependent disease risk mechanisms and studies on the clinical return of individualized polygenic disease risk. Her talk today is titled The Relationship Between Genetic Variants That Regulate Blood Eosinophilia and Allergic Disease Risk. Leah, welcome.
Thank you so much. Please current share. Share. Is this sharing appropriately yet?
Yes.
Great, thank you. So today I'm really happy to join you again for those who were with us in Montpellier and for the first time for those who weren't able to make that trip. I learned so much in the last IES meeting, and I learned even more today from our last two speakers. My talk's going to be a little bit different. I'm really going to try to highlight the future for using genetic markers to understand eosinophil biology and the relationship with eosinophil biology and other allergic diseases. I'm also going to attempt to highlight some resources that are publicly available. I'm going to give you some URLs that you can type directly into your computer so that every single person on this webinar today can start investigating the genetics of peripheral blood eosinophilia based on a couple of resources I'll walk you through. First, I'd love to just introduce myself. I have a laboratory on the bottom that I share with a computational biologist, Dr. Matt Weirach, and together we study the genotype-dependent mechanisms of disease risk, especially around immune diseases. And we do so at Cincinnati Children's, and our research building is shown up top. A very large part of our lab is really focused on taking samples from patients, samples from people who we know the DNA sequence of, and really understanding what's happening in the context of functional genomics. So measuring the chromatin accessibility like you just heard about, or where are transcription factors binding, using tools to understand genotype-dependent regulatory activity. And then we try to take these measurements and make models of what's happening biologically. We use these models to make predictions that we test experimentally. We try to refine our models. And the goal of all that we're doing is to really improve diagnostics and therapeutics. Another part of our lab that's very relevant to today's talk is our work in the primary care setting of trying to understand how to get polygenic risk scores into a setting that they haven't been historically. So today we're going to talk a little bit about polygenic risk score development. In the study that I'm showing on this slide as a summary, over the last 6 years, we, as a consortium of 10 sites across the United States, through an NHGRI consortium called eMERGE, recruited 20,000 adults, 5,000 children, and developed polygenic risk scores for 10 different diseases, and pragmatically tried to figure out how do we get those scores, the results of those scores, into a primary care setting such that a primary outcome, our clinicians would do something differently, and secondary outcome, our patients and patient families would take the physician up on that recommendation. As part of this study, we also have deep integration with our community advisory groups that help shape how we think about using genetics in a primary care setting. But first, let's talk a little bit more about polygenic risk scores. So these are scores that take an estimate of an individual person's genetic burden of variants for one trait or one disease. Generally, polygenic risk is for diseases that have, and for phenotypes that have more than one place in the genome that changes the expression of that phenotype. There are some diseases and phenotypes that one place in the genome, if you have a mutation, you can predict with high certainty, precision, and recall that you're going to get that phenotype or disease. But for most diseases, like most allergic diseases, like peripheral blood eosinophilia, we know that there's many places across the genome that each, in a little bitty way, tweak the phenotype. So for a polygenic risk score, we can present this burden score as one of 3 ways. The most common way is as a percentile rank, saying that if we looked at the whole population, this person is at the 72nd percentile, this person is at the 10th percentile. Sometimes we also present the data as a measure of effect. So if you are in the top 10%, maybe you have a 70% increased risk for a disease, or a 50% increase in average eosinophil count, for example. In a best case scenario, we have enough epidemiological data that we can take these polygenic risk scores and we can get them into an absolute risk perspective. For example, the BOTA-SIA is a great example of a score for breast cancer, whereas the eMERGE consortium was able to add polygenic risk to that and tell people based on their age what the likelihood is that they would develop breast cancer within a specific period of time. As one of the learnings from my work in polygenic risk score space and our entire consortium's learnings is that really clinicians and patients highly prefer to understand their individualized absolute risk. What does that number mean for me in the context of a readout that is requiring additional research? And so usually polygenic risk scores start as a percentile rank and evolve as more data becomes available into relative effect and absolute risk. Importantly, the polygenic risk score is not a definitive test. It is not going to say that somebody has a disease or doesn't have a disease, or will have high eosinophilia or not have high eosinophilia. Rather, it's a distribution that has a biomarker type effect size. So DNA you're born with, it's one of your first biomarkers of life, and based on your genetics, we can tell what the rank order, for example, is of someone's genetic risk. In this graph that I'm showing on the right is an example of a dichotomous polygenic risk score where you have either yes, no, you have a disease or you don't have a disease, you have asthma or you don't have asthma. And you can see that people in the 10th quintile, people in the 90th to 100th percentile have about a 6-fold increased risk compared to the people in the 0 to 10th percentile. Importantly, this is not redundant with family history. There's a beautiful paper in the American Journal of Human Genetics that really pulls apart for diseases including asthma and atopic dermatitis and some other allergic diseases, that your family history overlaps with your genetic risk and your polygenic risk, but is not fully redundant, mostly because your family history also includes a lot of environmental data. Now on the right, I'm showing an example of a quantitative polygenic risk score. So for example, when we're making these scores for things like a trait that can be measured and it can have many different measurements over a continuum, for example, blood eosinophilia, you can imagine a score where the top 10th percentile have on average 300 extra cells per microliter in the blood. And that's an example of polygenic risk scores that have been developed that I'll present some of at the end of this presentation for many measurements of blood cellularity, including eosinophilia. We recently, with Mark Rothenberg, did a study of a disease called eosinophilic esophagitis, where we developed polygenic risk scores for eosinophilic esophagitis. And I thought I'd walk you through how we did that, because I'm also going to show you how some of these polygenic risk scores correlate together, and we can start finding patterns of, if you're at high risk for eosinophilic esophagitis, for example, you might be at have a genetically increased burden of eosinophils in your peripheral blood. So for this study, we took people with and without eosinophilic esophagitis, and we did a genome-wide association study. And we did that study in two different ways. First, on the left, we did it in the standard case versus control method. On the right, we also applied a multi-trait method that I'll walk you through in a couple of slides that allowed us to leverage the genetic similarity between allergic diseases and increase our statistical power to find associated genetic variants that were important for multiple allergic diseases and eosinophilic esophagitis. Of our outcomes, we certainly found eosinophilic esophagitis risk loci, which I will share with you, but we also developed this polygenic risk score, which will become more important. So the graph that we're looking at right now is called a Manhattan plot. It's called a Manhattan plot because you see these skyrockets of genetic variants that remind us of the skyscrapers of Manhattan. On the x-axis at the bottom, every other color is a different chromosome that is an autosome, meaning it's not one of the XY sex chromosomes. All the dots are graphed as a function of their statistical association with eosinophilic esophagitis on the left, which is graphed as a negative log p-value. So the taller the dot, the more statistically associated it is with the disease. This red dashed line shows us our threshold for saying that something is important for eosinophilic esophagitis. And you can see from this particular plot that we have plenty of dots on top of the dashed line, and we establish these new genetic risk loci. As I alluded to earlier, we also leveraged a new strategy that allows us to take advantage of the fact that many allergic diseases share a common genetic etiology, where we've also published a nice review of that earlier this year in JACI. And based on that, we were able to leverage the ability to take advantage statistically of that shared genetic etiology to find even more loci that are important for eosinophilic esophagitis, many of which are shared with other allergic diseases. So now we've established a lot more new EOE risk loci, far more than we've ever understood before. So how did we make the polygenic risk score? Well, the first thing we did is we took our results from the Manhattan plot, as you can see on the left, and we constructed a score. We chose to use a non-filtered method, although there are filtering methods that you can use. We found the non-filtering method to be the most robust. And we basically, for every individual, used a Bayesian framework to add up all the risk variants and then weight every variant for every individual person based on the effect size, so how much that variant increased risk of EOE. The next step was a development step. So when you're developing a polygenic risk score, you take a cohort that was not used to find genetic association, because that'd be circular logic, and you take that cohort, you split it apart, and you use part of it to understand how your polygenic risk score is performing and tweak that, that initial polygenic risk score. And then you use the second set to validate the score and make sure that the score is predictive in a cohort, again, that was not used to find the initial association. You'll note that there's different ways to evaluate these scores. Early on in polygenic risk score space, people like to use area under the precision recall curve, which is really a measure of a score's sensitivity and specificity for diagnosing or predicting a disease. In line with what I presented at the very beginning of this presentation, we now understand that these scores are more going to act like biomarkers. And so we choose performance metrics that align with that expectation. So we're looking at odds ratio per standard deviation or per decile, or increased risk per decile. And risk stratification is also something that we consider. So the first results I'll show you I'm gonna just acknowledge from the beginning, are highly unsatisfying. The people without EOE are in red, the people with EOE are in blue, and you'll note immediately that no matter how we construct that score, on the left with just our standard GWAS approach, and on the right with our more multi-trait approach, the curves overlap. And so if you were right in the middle of the polygenic risk score, it is people that have the disease have the same score as people without the disease. Importantly, it's non-intuitive, but the places you're going to learn the most about disease risk and risk of differential, for example, eosinophilia, is going to be at the tails. And by that, I mean on the edges of these distributions, where people, for example, at the -3 of this polygenic risk score almost certainly do not have EOE, and people at the 4 of the polygenic risk score almost certainly do have EOE. We can show that in a different way. So we change our graphs a little bit, and we show it as the increased risk compared to the first decile of each of the deciles. The bottom line on this graph is shown with the polygenic risk score that we made using just a standard genome-wide association study approach. The top line is the study that we used, where we leveraged the increased power we got from the shared genetic association or heritability across allergic diseases. And you can first see that on the y-axis, we're looking at the increased risk of getting EOE. So for example, at the 10th decile, so these are people who are in the 90th to the 100th percent of genetic risk of disease. Based on the score that we developed with the standard GWAS approach, these people have a 5-fold increased risk compared to people in the 0 to 10th decile. With the MTAG approach, this multi-trait approach, we're at a 11.5-fold increased risk. Well, now we're getting into a space where knowing that you're in that 10th decile is actually even as predictive, for example, as knowing that somebody smoked and their risk for lung cancer, or knowing the impact of, for example, having high eosinophilia is for predicting that somebody is going to have certain allergic diseases. This is where everything gets really interesting. So now that we have a polygenic risk score that is specific for the burden of eosinophilic esophagitis, we can take that one score that we have for all the people in, for example, an electronic medical record system from that eMERGE study I discussed, where we have hundreds of thousands of people who have hundreds of thousands of medical records. And we can say, based on the score of each of those hundreds of thousands of people for eosinophilic esophagitis, what other diseases are they likely to have? The first thing that pops up is eosinophilic esophagitis. Well, that's just a replication of the fact that our score is working. So we'll just take that as a positive control and not celebrate ourselves. But then you start looking at what other phenotypes are coming up out of the electronic medical record, and note that the first one is the eosinophil count. The way you interpret this is that your genetic burden of eosinophilic esophagitis is predictive of having a higher peripheral blood eosinophil count. This is not hypereosinophilia. This is not a disease. This is just your CBC-based eosinophil count. You'll also note that it's associated with allergic reactions to food. And if you go down a little bit further, you'll find other allergic diseases. You'll find the Latexitive ligament and hypermobility syndrome, noting that people with hypermobility syndrome are far more likely to have eosinophilic esophagitis than somebody who does not. And this particular analysis indicates that there might be a genetic basis for that enrichment of eosinophilic esophagitis within this cohort of patients. This same strategy can be used for other polygenic risk scores. Okay, here's where if you're on a computer, I encourage you to take a screenshot, to grab a piece of paper and write down these URLs, or to I'll even put them in the chat afterwards, but you can Google GWAS catalog and get to the resource that's on the left. You can Google polygenic score catalog and get to the resource that's on the right. These are two incredible resources that I'll spend a few slides showing you what is in these around eosinophil biology that I have not seen a lot of work come out of that is more specialized and specific to these resources, but they're ripe for investigators to dive into to really understand the genetic basis of the variance in the number of eosinophils in the peripheral blood across a population, and understand how that might help us learn something about eosinophil biology. And I'll give you some examples. One thing I'll just say from the beginning is that there are both genome-wide association studies and polygenic risk scores that have been developed for eosinophil counts in the blood, eosinophil percentages, different measurements such as the density based on forward scattered, side scatter, the calculation of what percentage eosinophils are as a proportion of leukocytes, and other more protein-specific measurements of eosinophils. So all of those scores have been used in a genome-wide association study. The data are there and ready, and polygenic risk scores have been developed for these top two, eosinophil count and eosinophil percentage. In fact, there's 101 studies of peripheral eosinophil counts in the GWAS catalogs across many different databases, not just the UK Biobank, but also other biobanks from Finland, from Japan, from other just big resources, all of us. The vast majority are based on biobanks that also connect with electronic medical record data, such that you can not only understand the genetic basis of eosinophilia based on the people in the study, but then you can connect that with the other phenotypes and clinical presentations that these people are having in the clinic. There are obvious limitations to just taking off-the-shelf genetic studies of peripheral eosinophilia. One, I did not do anything to go in and curate these to account for parasite infection. I didn't average eosinophilia over a time point, noting that most of these people have many different measurements of eosinophilia, and this just took the top one. And I did not go into this resource and do the work that would be needed to remove people, for example, who have hypereosinophilia. So those are all opportunities that I hope someone on this call might jump in and do an even more refined study of. But even without addressing those limitations, peripheral eosinophilia is associated with genetic variants near really biologically important genes for eosinophil biology. I'm listing some of the ones that caught my eye on this slide, and I'm going to do a deep dive in the next two slides over some specific findings for CCR3 and AL18RAP and AL1RL1. So we know that that CCR3 is an important chemokine receptor that helps eosinophils travel towards eotaxins 1, 2, and 3. There's a genetic variant in CCR3 where the G allele is very rare, meaning 0.1% of chromosomes, 0.4% of chromosomes of African ancestry have this rare variant that leads to a large decrease in eosinophil count per G allele. This variant changes an amino acid in CCR3, and in a model of proteins from polyphen 2 is predicted to be likely damaging. This variant also, if you look at people with this variant, they tend to have much less eosinophilia in their peripheral blood. So that's one example. Another example that just pops out of these resources is from a loci that is near two genes that are right beside each other, AL18RAP and ALRL1. This variant happens to also be consistent with a pretty substantial, not as substantial as the CCR3, it's more common, increase in eosinophil count, and it leads to a decrease in the receptor for IL-33, which we heard about the importance of in eosinophil development from our first two speakers.
Yeah,
And
I'm sorry to interrupt. I just want to double check on the time for you.
I am almost done. I will.
Excellent. Thank you.
Pop on. So basically, we have an eosinophil polygenic risk score that was developed and put into the supplement of a paper that has not been, there was no other follow-up within that paper. This paper developed about 100 polygenic risk scores. From that polygenic risk score of eosinophilia, you can see that the polygenic risk score for eosinophilia actually correlates with other allergic diseases, including our work from Eosinophil-Associated and some work that we publishing a polygenic risk score for asthma. But it's not associated with all allergic diseases. For example, it's not associated with the polygenic risk score for atopic dermatitis. So the summary of what I've shared today is that these polygenic risk scores are really important tools for collapsing an individual's genetic burden for any phenotype, a disease, or something like peripheral eosinophilia into one number, and that large biobanks have performed training and testing for many of these scores that really have been unstudied and undeveloped in a biological sense. They're really exciting opportunities for eosinophil biologists to really seriously engage with the genetic basis of eosinophil phenotypes. And in the future, polygenic risk, clinical presentation, environment, and other biomarkers might well come together to help clinicians shape the way we treat patients in an individual manner. And I'll just end by thanking many people, especially Mark Rothenberg, who I've worked with for 2 decades, as well as others who've contributed to any of the work I shared today. So thank you.
Thank you. That was a fabulous talk. I really appreciate it. We don't have time for questions. However, I am going to sort of like lob one at you anyway. Maybe you could answer it in the chat afterwards. So your talk was really focusing on the DNA that we inherit. How does that interlie or weave with maternal fetal interactions and DNA methylation and all the other things that we know that you can inherit, that one way or another, not necessarily directly from your genes, but modulation of your genetic expression, and how would that impact your risk scores? So if you could sort of put a note about that in the chat, I would really appreciate that. So, but thank you very much for that fabulous talk. And my colleague is going to close us out this morning. So thank you.
Yeah. Well, thank you everyone for attending the webinar and for your questions during the discussion. The recording of this webinar will be available on demand. You will be able to find it on the International Eosinophil Society website. I would also like to make you aware that all attendees will receive an email asking you to complete a post-webinar evaluation. So please look out for this. We very much value your feedback. And finally, I would like to thank again our 3 fantastic speakers for taking the time to share their work with us today. So thank you.
Thank you also to the speakers and to Nicola. Thanks.